intense generated by radioactive decay and heavy bombardment melted much of the early Earth. This molten stage allowed the planet to different.
那个鼠标指针给它调出来。
Okay.
对对。
The molten stage allowed the planet to differentiate with denser materials like iron and nickel sinking to form the core, and the lighter silicates forming the melting and the crust. In other words, this very beginning sets the stage for everything that came later, including the chemistry, and ultimately gave rise to life. After Earth's beginnings with intense heat and from impacts and radioactive decay, the planet gradually started to cool down. As the surface temperature dropped, volcanic eruptions released large amounts of water, vapor, and other gases into the atmosphere. Eventually, this vapor condensed and began to fall as rain, a process that lasted millions of years. Over time, the accumulated rainfall filled low-lying areas, forming the first oceans. Early oceans were likely hot and filled with dissolved mineral, creating what many scientists call the chemical cradle of life. In other words, while the formation of Earth provides the physical foundation, the cooling and appearance of oceans provide the environmental stage, where life is chemistry could began. As the ocean formed and the planet continued to cool, another major transformation was happening. The development of early Earth's atmosphere, unlike today, the earlier atmosphere contained no oxygen. Instead, it was dominated by gases released from volcanic outgassing, such as carbon dioxide, nitrogen, water vapor, and small amounts of methane and ammonia. This period is known as Helden Eon, when Earth's surface was still hot and unstable, constantly bombarded by asteroids and shaped by volcanic activity. These gases created a weakly reducing atmosphere, meaning it could support chemical evolution. The next key question is what provides the energy to drive the first chemical reactions of life? Several natural energy sources were available on the early Earth. Lightning could have sparked reactions in atmosphere, forming simple organic molecules. A concept later tested in the famous Miller experiment. Ultra UV from the young sun also played a major role, since there was no ozone laser to block high-energy light. And deeper within the planet, volcanic and geothermal heat powered chemical reactions around hydrothermal systems. Together, these energy sources create a highly dynamic and reactive environment, one where basic molecules could assemble, transform, and eventually evolution give it rise to more complex organic chemistry. It is worth noting that the plasma environment is very important. it is known to us that more than 99% of the observable matter in the universe is in a plasma state. So what is plasma? Plasma is a high temperature iron gas. When the gas is heated into a sufficiently high temperature, atoms lose electrons and become a mixture composite of positively charged ions and negatively charged free electrons. Because these charged particles can move freely, plasma flows like gas and conducts electricity like an electric body, and is called the first state of matter. The most famous plasma discharging experiment is the Miller experiment. Miller was the pioneer in the study of the origin of life, who was the first to achieve the transformation of inorganic molecules into amino acids through discharge. So discharge can generate high energy electron and highly reactive free radicals species on the chemical bonds between the molecules and generate new molecules. Thus, plasma discharge scenarios are of great significance for promoting the origin and the evolution of life molecules. In recent years, researchers have begun to simulate the early Earth and even extraterrestrial plasma scenarios in laboratories in order to explore the origin of molecules. Unlike traditional spark discharge experiments in gases, these studies focus on plasma interacting directly with liquids, which is a system that better mimics the dynamic interface between the early atmosphere and the primordial ocean. The left side is a cloud discharge on the liquid surface, which was published in PNAS. The experiment shown on the left demonstrates cloud-like plasma discharge formed above a liquid surface containing water and dissolved gas such as nitrogen and carbon dioxide. When the high voltage sparks around 10 to 40 kV voltages are applied, a visible plasma forms in the gas phase and interacts with the liquid below. These interactions drives a variety of redox and radical reactions. The right side shows the formation of primordial cell-like structures, which was recently published in PNAS 2. The study on the right takes this concept even further. Researchers found that during plasma discharge in liquid environments, micrometer scale structures spontaneously appeared, showing in the SEM images. This cell-like structure exhibit organic-rich composition, mostly carbon and silicon, and as shown in the EGX spectrum. So what's remarkable is that these structures not only form spontaneously, but also show membranes like boundaries, possible analogue to protocol cells. This suggests that plasma discharge could not only synthesize organic molecules, but also drive self-organization into primitive cell-like structures. Together, these two studies represent a major step forward in our understanding of how energy-rich plasma environment would have contributed to the both of chemical and structural origins of life. Our recent research has also revealed that the underwater plasma bubbles can efficiently promote the formation of life molecules. Research has found that among the 12 amino acids and 3 bases detected, the contribution of bubble plasma is several times higher than that of needle liquid. Another important aspect is the interface environment. This interface provides conditions that are conducive to molecular evolution. The left image shows the gas water interfaces, such as bubble effects in hot springs. Studies have shown in the bubble walls can enrich and promote the formation of nucleic acid and liquid protein particles. And the mineral water interfaces is also important, which showed that minerals not only catalyze synthesis reactions, but also provide structure templates. And finally, the wet-dry interface is also very important for the chemical evolution. So far, we have explored the well and underwater conditions that might have originated from deep sea vents to volcanic conditions and plasma environments and so on. But another crucial question remains: how did non-living chemistry gradually turn into living system? This brings us to the concept of prebiotic chemistry. The stages that bridge geology and biology. It focuses on the chemical evolution that occurred before life began, when small molecules like gases were transformed into organic building blocks like amino acids, nucleotides, and lipids through energy inputs such as lighting, UV radiation, and catalyzed surfaces. These molecules could react, linking together, and eventually form more complex molecules, such as proteins, RNA, and other structure. In essence, prebiotic chemistry presents the important slow transition from simple molecules to first protocells, marking the chemical foundation of life's origin. Now that we have introduced the concept of prebiotic chemistry, the chemical evolution from simple inorganic molecules to building blocks of life, let's take a closer look at the first step of this process. In this stage, scientists focus on how simple gases present in the early atmosphere, such as CH4, NH3, and water vapor, and so on, and carbon dioxide, how they could interact under different energy sources to produce organic molecules, particularly amino acids. Various environments have simulated these early conditions, including electrical discharge, shock waves, high temperature heating, and plasma discharge, and so on. So here we looked at the first step of chemical evolution, and how simple gases like ammonia and water could form basic organic molecules, such as amino acids on different energy sources. Here we showed the various pathways to the basic molecules from gases. For amino acids and peptides, the Strecker reaction and the various Strecker reaction is very important. from the to synthesize the amino acids and the peptides. Some conditions like plasma discharge and wet dry conditions could be stimulated to synthesis. And nuclear bases like adenine could be synthesized from HCl polymerization by oral. And sugars, it is shows that Formo's reaction is a very important pathway to synthesize the sugars and the lipids. In the molecules such as the amino acids, nucleic acids, sugars could form through various prebiotic reactions. But the university raised an important question. Was there a single and simple molecule that could serve as a common chemical starting point for many of these pathways? Recent research points to formamide as one of the most important molecules which could promoting catechase. Formamide is a small, highly reactive molecule that can act both as a solvent and as a reactant. Under different energy sources, such as UV light, plasma discharge, or mineral catalysis, formamide can yield a wide range of key biological precursors. As shown in the figure, formamide chemistry can transform into amino acids and nucleic acid bases and sugars and so on. So far, we have seen how simple molecules on the early Earth could give rise to a variety of life essentials, building blocks, amino acids, sugars, and other molecules. But the next major question is, how did these individual components come together to form a system? One leading hypothesis is that addresses this transition is the RNA war, which was first proposed by Walter Gerber in 1986. It suggests that before DNA and protein appeared, early life forms were based primarily on RNA. RNA is unique because it can perform two critical roles. The first is it can store genetic information, serving as a template for replication, and the second is it can catalyze chemical reaction, acting like an enzyme and ribosome. This dual capability means that RNA could have driven both heredity and metabolism in early life systems. Over time, as complex increased, proteins took over and most catalyzed functions, and DNA became the main genetic material. But the RNA water represents that crucial intermediate stage connecting chemistry to biology. In addition to RNA water, some scientists propose that before complex biopolymers exist, it may have originated from a metabolism first scenario. This brings us to the new hypothesis, or the ion sulfide wall hypothesis, proposed by Gunter around the year 2000. It suggests that life began on the surface of metal sulfide minerals, such as some sulfide minerals. well self-sustaining chemical cycles could form. This mineral surface provides both energy and catalyzed sites, allowing simple inorganic molecules like carbon dioxide to be transformed into small organic compounds, for example, amino acids and peptides. So, in prebiotic chemical reaction networks, it is very important to the biological systems formation. This brings us to the concept of prebiotic the chemical reaction networks funding in the early Earth. Rather than focusing on the synthesis of a single compound, scientists have now exploring how large numbers of molecules could have formed and interacted under prebiotic conditions. As shown here, recent work and some molecules of modern metabolic cycles, like the reductive citric acid cycle, can exponent roles emerge from simple chemical building blocks. These interconnected reactions could have provided a self-sustaining. Now, we come to one of the most fascinating and still unsolved mysteries in the original plant: carity and biological selectivity. So what is carity? A molecule is carrier if it cannot be superposed on its mineral image. And the question here is, why does life use almost expressly one-handedness? It is a still unsolved and open question. Scientists have proposed some possible mechanisms, including physical chemical breaking and surface mineral templating, like autocatalysis amplification. Once a site exists, reactions favor that handedness. Here is some recent advances. The scientists found that in a peptide ligation, there was an initial preference for heterochiral ligation over homochiral ligation. However, further analysis reveals that this can actually lead to carrier amplification. That is, even if only a small portion of the carrier basis is initially achieved, it can be amplified to a high purity of the same carrier chain through compactive reactions. Another advantage is that the scientists found under simulated prebiotic conditions, the protein amino acids can self-activate and be linked into peptides through peptide bonds. The linking processes shows significant and high isochiral peptide generation. The past provide a new chemical mechanism for how to achieve carrier peptide in a water environment. Now let's talk about one of the most central molecules connecting prebiotic chemistry to modern biology, the ATP. ATP is often called the energy currency of life because it stores and transfers energy within every living cell. Structurally, ATP consists of three main parts. nitrogen bases, adenine, a sugar ribose, and three phosphate groups connected in a chain. The bonds between these phosphate groups, especially the terminal phosphohydride bonds, are high energy bonds. When one of these bonds is broken, ATP is converted into ADP and inorganic phosphate, releasing energy that drives countless biological reactions. In modern organisms, ATP is the power for the living, and it powers nearly all vital processes, such as biological function, providing energy, and mineral transduction, materials transformation. So it is worth noting that the ATP could be simulated formation synthesis in the early Earth. It links the chemistry of mineral phosphates and prebiotic energies transfer into the evolution of biological metabolism, the synthesis of ATP. It is also possible in the early Earth. In the previous slide, we look at ATP and the energy currency of life, showing how philosophers is central to energy storage and transfer. But phosphorus plays another equally fundamental role. It provides a stable backbone for molecules like DNA and RNA. This idea was elegantly extended by Prof. Frank Wismir, a pioneer in bioorganic chemistry. He emphasized that phosphorus is not only reactive enough to support energy metabolism, but also exceptionally stable when incorporated into biological polymers. Here you can see that the phosphodiester bonds connecting nucleotides in DNA and RNA are remarkably resistant to hydrolysis. The half-life of phosphate bonds in DNA can reach around 10,000 years under natural conditions. That means once formed, these molecules can persist long enough for genetic information to be stored, transmitted, and evolved, which was critical for the emergence of life. If we compare phosphorus with other possible elements like silicon or sulfur, you can see from this table that non-come closing stability. And now that we see how phosphorus provides both energy and stability for life molecules. Here, we take a close look at another fascinating aspect: phosphorus chemical selectivity in the evolution of biomolecules. This table summarizes experiments and theoretical analysis showing that phosphorus specifically favors alpha amino acids. The very type of amino acids used in modern proteins. You can see three types of amino acids here: alpha, beta, and gamma. You can see three when we compare how these react with phosphorus containing compounds, only alpha amino acids formed stable phosphorus-mediated bonds, shown in the red. Now that we have seen how phosphorus chemistry helped shape the selection of alpha amino acids, the next great question is, how was these amino acids linked to genetic information? This takes us to one of the most groundbreaking experiments in molecular biology, the origin of genetic code in 1961. Marshall and Nierberger and Marshall conducted a simple yet revolutionary experiment. They used a cell-free extract, basically biochemical soup containing all the components for protein synthesis and added two key ingredients, ATP and polyU and synthetic RNA. To their surprise, the system produced a long-chain made entirely of the amino acid PHE. This demonstrates that the RNA trapped UU codes for PHE, marking the first described codon in the genetic code. This discovery opened the door to understanding how specific nucleotide in RNA respond to specific amino acids, the foundation of universal genetic code we use today. It transformed our understanding of life's molecular language, linking information to function for the first time. So this experiment essentially revealed the bridge between chemistry and biology, how the random molecules of early Earth became organisms into coded replicating systems. Nature established a perfect carrier partnership, L amino acids for proteins and denucleosides for nucleic acids. And the ribosome acts as a gatekeeper that enforces this molecular rule. Based on this, our team proposed to establish the chemical recognition of early amino acids and nucleosides in order to explore the origin of the genetic code and sensitively propose the chemical models for the origin of the genetic code in both the organic phase and the aqueous phase environments. Further, it has been observed at the molecular level that the amino acids and the nucleosides have inherent carrier selectivity in the process of forming amino nucleotides. Denucleotides are more likely to react with L amino acids formed from the corresponding amide nucleotides. For L nucleotides, D amino acids are preferred. That there may be a carrier instead of a carrier with Earth system, further suggests that there may be a life into the mirror of the world with Earth. So here is the outlook. Here is a question. In the next 20 to 30 years, might we find the life beyond Earth, or at least strong evidence of it? And how does the evolutionary process occur in the universe? So the question, how to explore it? Here summarize the 4 points. The one is carrying out experiments in simulated extraterrestrial environments on the ground. Second is exploration of origin of life on the space station. Observation of chances of life by space telescopes. And finally, maybe some sampling of exterior asteroids. And thank you for your support of the National Natural Science Foundation. And thanks for my master supervisor, Prof. Yufeng Zhao, and my doctoral supervisor Ren Zhou and my teacher Jian Xi. And thanks. Okay, there is any question?
Any question?
对，要共享哦。对，
这个样子。
哎，好，你再共享。先打开你的，先打开你的 PPT。没共享过，共享，对不对？共享，选到你们看。
मैट्रिक से पहले का है
鼠标给它弄出来，鼠标那个激光笔，对。
So good afternoon, ladies and gentlemen. Thank you for your time this afternoon. I'm very happy that you come to this Ningbo University COMSTECH joint program in Aerospace Biomedicine, Pharmaceuticals, and Artificial Intelligence. First of all, I'd like to introduce myself. My name is Chen Sude, and I'm a postgraduate student at Ningbo University. So it's a great honor for me to indeed to be here to share with you the topic of prebiotic chemistry and the origin of life. So during my presentation, I will give a brief introduction to this discipline. Mr. Gan has introduced this topic in another perspective, so I will show you another some tips or some new outlook. We are delving into the question of how non-living matters give rise to the first living system. To start off, let's reconsider these questions that are put on the screen. Are we alone in the universe? How and where did the life on Earth arise? In fact, these two questions were from a special issue of 125 questions, What We Don't Know, published by Science magazine 20 years ago. The question to the origin of life has existed since the beginning of the recorded history. To understand them, a most fundamentally question should be claimed, that is, what is life? So understanding the framework of the question, what is life, is a fundamental to formulating other questions that are central to prebiotic chemists, such as where else may life be found and how do we search for life elsewhere? So definitions of life can serve as essential guide when searching for life beyond us. The concept and definition of the life largely depends on its historical context, which full definition can be traced back to ancient Greece and Aristotle, where the concept of psyche or soul was used to describe the properties unique to living things. These definitions changed drastically starting with Enlightenment, starting with quasi-mechanical definition by Bacon and Descartes in the 17th century, followed by Schleiden and Schwann's focus on the cell as the unit of life, and finally definitions based on evolution by Darwin, Lamarck, and others in the 19th century. In the early 20th century, models of the life turned on the origin of life questions, founded in new knowledge and discoveries. First, Haldane and Oparin developed theoretical descriptions of life origin, and Miller and Urey provide experimental evidence to support the abiotic origin, as Mr. Gan introduced. With the discovery of DNA and its composition and structure by Watson, Crick, and Franke, information storage was added to the perspectives and definition of life. With the advent of the digital era now, definition of life has become more abstract, specifically considering functions rather than form, such as the storage and transmission of information, the complexity of the system, and metabolism. So there's no current consensus on the definition of life. Historical perspective has emphasized the functional features, such as chemical interactions and provide energy, that is metabolism, or the transmission of information, for example, actions, and use a generalized feature that are thought to be common to all life. The working definition of the life that NASA uses is a self-sustaining chemical system capable to the Darwinian evolution. So life on Earth is known as the terran life. Although terran life is remarkably diverse, all organisms use the same molecular building blocks in roughly similar proportions and share similar metabolic pathways. The commonality of these elements can be explained by a shared ancestry that unites all life on Earth. So the elements carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, known as COMSTECH, are essential components of all cellular biomolecules. And all known organisms on Earth rely on the same fundamental set of biomolecules to carry out the function of life, including nucleic acid, proteins, lipids, and polysaccharides. And then common process and properties shared by all known life include order structure, reproduction, metabolism, response to the environment, homeostasis, and evolution. So the first thing is a cell as a unit of life, and life except for virus on Earth is cellular, either as a single cell or multicellular unit. The cell provides separation from the environment and enable life to maintain stable conditions inside the cell to support survival. Every cell on Earth is composed of networks of biomolecules that keep the cell alive. So cell as a host for genetic replication, bodies for unicellular organisms, an essential component for multicellular organisms. The second thing is the elemental building blocks of life. The elemental composition of life on Earth informs the understanding and search for life elsewhere. So water is universal and predominant in all life forms, and is a critical medium for metabolism reaction. Including water, about 97% of mole of cellular mass is composed of these 6 elements, is COMSTECH. While the remainder is various amount of sodium, magnesium, chlorine, potassium, calcium, magnesium, iron, and cobalt, copper, and zinc, molybdenum. They are micronutrients, and to an even lesser degree, which is silicon, vanadium, and nickel, and tungsten. They are trace elements. So COMSTECH form the basic structure of all biomolecules and larger polymers, where the less common elements facilitate the solubility or catalytic properties of these molecules, maintain ionic equilibrium, and regulate redox reactions and membrane potentials. So the abundance and chemical property of water makes the ideal solvent for the terran life. Water has a naturally asymmetric charge distribution that results in the permanent electric dipole, with partial negative charge on the oxygen and the partial positive charge on the hydrogen atoms. This dipole allows water to remain liquid over a relatively wide range of temperature, and also act as a both H-bond acceptor, hydrogen bond acceptor, and a hydrogen bond donor, where the hydrogen is a H-OH bond. The weak charge in water allows it to dissolve ionic compound. So these characteristics make water crucial for many of the fundamental redox reactions of life. And water is also unique compared to other solvents in that it expands upon freezing due to its strong hydrogen bond, and will float to the surface of liquid water, causing bodies of water to freeze from top down. The top frozen layer insulates the liquid beneath, preventing deeper zones from freezing completely. So this unique feature would have enabled the survival of oceanic life during snowball Earth events, during which the entire planet have been covered in ice, and also help to explain how liquid oceans can persist on Europa and Enceladus, which is the moon of Saturn. And now let's focus on carbon, hydrogen, oxygen, and nitrogen. So carbon forms the backbone of virtually all biomolecules, all that bacteria life. Its capacity to form 4 stable covalent bonds with a wide range of elements, resulting in diverse and complex micro-scale structures. So gaseous CO2 carbon dioxide in the atmosphere serve as a primary source of carbon for life, as the primary source on Earth today. So it's converted into various organic molecules, either use energy from light via photosynthesis or by manipulating redox potentials through chemical reactions, which means chemosynthesis. These molecules can then be incorporated by organisms that do not produce their own organic carbon. And atomic hydrogen and oxygen can be derived from breaking apart water molecules and would likely have been readily available for incorporation into cellular materials on early Earth. Beyond their role in biomolecules, hydrogen and oxygen influence the redox potential of geological systems. They react easily with metals and other reactants and other redox-active species, often driving the key biological process. So in fact, hydrogen-containing gases are the most reducing and typically most reactive. For example, the ammonia, methane, it's sulfur, it's hydrogen sulfate and hydrogen. While the gases with oxygen are some of the most oxidizing. Contrastingly, while hydrogen and oxygen are abundant, most of the nitrogen on contemporary Earth exists as a chemical inert nitrogen gas, which is most stable. The triple bond that binds the nitrogen atoms given the nitrogen gas is highly stable. However, certain bacteria and archaea can convert it to most reactive molecule, such as ammonium ion, H4+, and nitrite, NO2-, and nitrate, NO3-. Abiotic process like the powerful electrical discharge from lightning can also oxidize nitrogen gas into nitrous oxide, NOx. This can eventually form biologically accessible molecules like nitrate and nitric acid. So phosphorus is an important component of biomolecules, including nucleic acid, lipids, and ATP, adenosine triphosphate. Almost entirely as a negative charge also phosphate. The phosphate backbone enhances the solubility of nucleic acid in water and prevents their leakage from the membrane, which restricts the diffusion of charged ions. In addition, the negative charge may be critical for the stability of nucleic acid when confronted with slight changes in their structure. So phosphate can have many roles in a cell, including serving as a directly phospholipid in the cellular membrane, which this one. It's a coenzyme A. An energy source by high energy bond pyrophosphate is a diphosphorus component with a POP linkage, or as a regulatory agent in protein activity, as a phosphate group added or removed from amino acid can cause a significant change in the three-dimensional structure of protein. So sulfur's ability to form redox-active disulfide bond between the two organic compounds has made it a fundamental part of terran life. In proteins, disulfide bonds form only between the two cytidine, but not the S-containing methylidine. Formation of disulfide bond is reductive dependent and impact protein folding and therefore the reactivity of protein. And sulfur can exist in many oxidation states, the -2, the 0, +2, 4, and 6. And it's an important source of energy for microbial life. Sulfidic compounds are also utilized by virtually as a terrestrial life to transport electrons in the cell via metallic clusters, for example, the iron sulfur clusters. So notably, the sulfur atom in cysteine often serves as a ligand in protein metallic cluster, holding the metal such as the iron or zinc in place. The importance of sulfur in many of life critical function has led to many suggestions that sulfur and iron sulfur clusters may have been critically important for the origin of life in early Earth environments. And life selectively requires smaller quantities of many other elements as well. The most prevalent being sodium, potassium, calcium, magnesium, and iron. These species tend to exist on Earth as ions and can generate potential difference across the membrane. So it can help stabilize the charged organic molecules in solution or even be used as structure components. Life also incorporates some transition metals, for example, the iron, magnesium, copper, titanium, cobalt, and zinc, which radically gain and lose electrons, catalyze reactions, and control electron flows in metabolism. So all known organisms on Earth rely on the same fundamental set of biomolecules to carry out the function of life. Cells store information about protein synthesis as the genes in DNA. The code in DNA is mirrored by messenger RNA, which detect the sequence in which amino acids are combined, in the ribosome to produce proteins. This typical flow information from storage with expression is DNA to RNA to protein is known in biology as a central dogma, and it's found across all of the terran life. So protein serves numerous functions such as reaction, catalysis, structural support, and electron transport. And lipid encompass a broad range of organic molecules, including phospholipid used to form cellular membrane, and triglyceride used for long-term energy storage, while cell use adenosine triphosphate ATP and nicotinamide adenosine dinucleotide NAD for short-term energy storage. And last, the sugars and polysaccharides composed of glucose and similar sugar and more densely packaged lipid are used for long-term energy storage. The long-term energy storage. And polysaccharide also commonly used as structure elements such as cellulose and structure components of the cell walls in plants. Chirality is the property of asymmetry where molecules, while having the same structure, are non-superimposable. Each chiral state is known as enantiomer. And no combination of translations or rotations of chemical bonds within an enantiomer will allow it to be perfectly superimposed onto its mirror image. Enantiomer can be either left, we denoted with L. It's a Greek word, which lexo means left, or dexo means right, right-handed, defined by the direction in which they rotate plane polarized light. A solution consisting entirely of one enantiomer is termed homochiral or enantiomer pure. When both enantiomers are present in equal concentration, the solution is rhythmic. If there is an excess of one enantiomer, the solution is said to be possess an enantiomeric excess. So chiral preference is seen as the fundamental property. A fundamental property of life. Indeed, all extant life forms exhibit a chiral asymmetry, namely nucleic acid exclusively employ D sugars, while protein relies solely on L amino acid. Also, although the enantiomers share identical physical and chemical properties, their unique spatial configurations lead to different outcomes when they react with other molecules. Thus, consuming the opposite enantiomer, for example, the L sugars or D amino acid, can be harmful to modern life. However, some organisms make use of opposite-handed molecules for specialized purpose. For example, the D amino acids have been identified in various cellular functions, such as structural support, defense, and cellular communications. So the following part, I will talk about the prebiotic process. Within the first billion years of the Earth's history, the planet transformed into a hot, barren, and inhospitable landscape to an environment conducive to the emergence and presence of life. Events following the formation of Earth around the young sun determined the chemical environment in which life may have originated. The earliest period of Earth's history is known as the Hadean Aeon, and is being generally taken as a point when Earth began to differentiate to form the core, mantle, and crustal layers. Around 4.56 billion years, the Moon was formed due to the impact between a Mars-sized planetesimal named the Theia, and the photo Earth within about 60 million years of Earth formation. So the impact would have produced enough heat to cover Earth in a magma ocean about 7 kilometers deep. This would have boiled off the oceans and atmosphere and sterilized the surface. Although the moon forming impact was relatively unlikely, it has important implications for Earth's habitability. The impact increased the abundance of heavier elements of the surface, while the moon itself creates the tide that helps cycle elements through the hydrosphere and atmosphere. And about 10 million years after the moon forming the impact, magnesium, iron, and silicon-rich olivine and pyroxene minerals crystallized to form a predominant crust. Exposure to water would have oxidized the elements of this crust to form serpentinite rocks and hydrogen gas, by a process called serpentinization. As the planet cooled, water vapor delivered by comets and asteroids would have condensed and rained out, lowering the temperature that triggered further condensation of a growing ocean. By the end of Hadean, global oceans are expected to have contained a greater volume than the modern oceans and been anoxic, enriched in ferrous iron and sulfate and dependent in sulfate. The atmosphere was also anoxic and is thought to have been composed most of nitrogen gas and carbon dioxide, CO2, with some hydrogen, water, hydrogen sulfide, H2S, and sulfur dioxide, SO2. This table summarizes the current understanding geochemistry on early Earth. So investigations into a region of life can be either top-down, it's biological approach, and bottom-up is a chemical approach. The top-down approach is reverse engineers the life into its most basic constituent to consider the potential materials and reaction outlets by the first life form. And top-down approaches are hindered by the dearth of a hidden geochemical record. And the bottom-up approach considers the fundamental physical and chemical mechanism of molecular system in the context of primordial geochemical environment to deduce how simple molecule building blocks develop into more complex biomolecular system. Experiments simulating early Earth chemistry together with meteorites and returned extraterrestrial samples suggest that early Earth posted a myriad of organic molecules derived from exogenous and endogenous, which means external and internal sources. So it's uncertain when life emerged on Earth. Evidence for the life may extend as far back as 4.1 billion years in the form isotopic ratio of carbon-13 in graphite that are consistent with having originated from metabolic activity, although this evidence is highly controversial. So energy is the critical ingredient for abiogenesis, as it enables the generation and maintenance of biological matter. The higher the amount of energy needed for a reaction to proceed, which is called activation energy, the less likely it is to occur spontaneously. So the activation energy can be lowered by a catalyst that can be lowered and itself will not consume, thereby increasing the rate of the reaction. So forms of energy are interchangeable. Let's take advantage of this to precisely control when and which chemical reactions taken place. The most prevalent example of this was life harnesses the movement of proteins to synthesize adenosine triphosphate, ATP, the primary energy molecules used by all terrestrial life. However, it is unclear whether the ability to harness and storage energy was necessarily for abiogenesis, or whether it emerged during life's origin. The prebiotic synthesis of AMP, ADP, and ADP, whether But geochemical gradient was vital to the initial creation of this equilibria, which could serve as a source of potential energy for the origins of life. The gradients occur between two adjacent environments which significantly different geochemical parameters, for example, the temperature, the pH, and salinity, that are separated by some sort of semi-membrane physical barrier that prevents rapid mixing and gradient dissipation, because they drive ATP synthesis in cells when cellular respiration. So pH gradients are the most vital for terrestrial life, as pH gradients are maintained within serpentizing alkene vents, which means hydrothermal vents. It might be possible that such systems were important for the emergence of resistance of life. And back to the elements, the most abundant elements within all life forms are CH and OPS we have seen before. Although the availability for prebiotic reactions on earlier would have varied significantly depending on the geochemical environment, the CH-ONPS species were likely relevant to the earliest form of life. A protein-protein chain reaction in the cores of low mass stars could generate light elements. The hydrogen fusion occur around the core, produce more helium that adds to the helium already into the core. Eventually, after about 1 billion years, the temperature of the core has increased to a point where it's possible for helium itself to fuse. So the temperature of the core increases to 100 to 200 million Kelvin, and this temperature is now possible for 3 helium nuclei to fuse together to form carbon and release energy. As a helium nucleus is also called an alpha particle, so this reaction is also called the triple alpha reaction. It proceeds from the 2 helium nuclei fusing together to form a beryllium-8 nucleus, which itself then fuses the helium nucleus to produce carbon-12. In high mass stars, the hydrogen fusion to helium is dominated by a more complicated cycle called the CNO cycle. Of all chemists occurring in space, the formation of carbon compounds is the greatest interest of researchers. By studying carbon compounds, we can get insight into whether the building blocks of life, such as sugars, amino acid, could have been synthesized in space and subsequently delivered to earlier. We can start to understand more generally what diversity of compounds can be formed in space. So once the precursors are formed, more complex carbon compounds, potentially the precursors to prebiotic molecules, can be produced. These reaction pathways have various names associated with them. Here just some of the variety of carbon reactions are illustrated that can lead to more complex carbon molecules, and particularly compounds with more than 1 carbon. The final pathway to compound with the multiple carbon atoms are still not fully understood. And the full diversity of carbon compounds observed in space have not been synthesized in the laboratory. However, the reactions above give us some ideas of the potential chemical process. These reaction schemes generate some of the vast array of carbon molecules, from the simple ones such as carbon monoxide to more complex compounds that have now been observed in space. You can see just some of the range of gas-phase carbon-containing molecules that are thought to have been identified. So prebiotic synthesis of primitive biomolecules as their monomeric precursors stems from the reactivity of CH-ONPS molecules that would have been readily available on Hadean Earth. However, as we discussed, the most stable form of these elements required for biosynthesis was relatively unreactable, oxidized molecules. Thus, a continuous source of reactive and reduced molecules was needed for high yield of biologically relevant compounds to have formed. This section will discuss several abiotic chemical mechanisms that may have been important to the origin of life. The separatization reaction is the hydrolysis of a mineral with a rock, generating hydrogen gas H2 as a byproduct. Separatization most often occurs within ultramafic minerals, such as common in Earth's mantle. And it may have also served as a source of hydrogen. On Earth or on other world, the production of hydrogen for ongoing subpotentization have been observed in the field, including at Lost City hydrothermal wind, Rainbow hydrothermal field, and the Oman ophiolite. Subpotentization generates OH minus, which is hydroxyl anions that would have resulted in a pH gradient with early Earth's slightly acidic waters, fueling carbon dioxide reduction to methane and potentially ATP. And then the striker synthesis occurs when ammonium, cyanide, and aldehyde combine to form an alpha amino acid. It begins with condensation of the ammonia and aldehyde to form an imine, which is then attacked by the cyanide anion. The cyano group can then be hydrated, generating an carboxylic acid. The striker synthesis have been perceived to have occurred in the primitive ocean and to have been the major source for amino acid within meteorites. So the reaction mechanism includes two parts: formation of alpha amino nitrite and hydrolysis of nitrite. So reductive amination is a process by which substituted amines, such as amino acid, can be generated via alpha keto acid. This reaction occurs relatively at low temperatures. For example, the room temperature to 70 degrees. And it's initialized with the condensation of an imine or ammonia onto an aldehyde or ketone to form an imine intermediate. That is then further reduced to the imine product. Iron is typically used as a reductant and water as a protein source. And the fourth one is the Fischer-Tropsch type synthesis. It can efficiently generate liquid hydrocarbons, for example, alkanes, alkenes, alcohols, from hydrogen and carbon monoxide in the gas phase. This process is performed with metal catalysis at high temperature and pressure. When conducted in aqueous solution in the presence of other gases such as ammonium or hydrogen sulfate, the reactions are known as Fischer-Tropsch type reactions. So such reactions have been proposed as potential abiotic source of fatty acid with up to 18 carbon long side chains, amino acid nucleobases, and hydroxyl acids, among other organic in various planetary environments, including with the deep subsurface of the crust, hydrothermal winds, meteorites, and the solar nebula. So FTT chemistry may have also been the source of organic matter found in the oldest Archean rocks, making it difficult to describe the geneticity of potential microfossils. So the fifth one is the Formose reaction. The Formose reaction describes the polymerization of formaldehyde, and is the leading mechanism proposed for the prebiotic origin of sugars, including ribose, glucose, and other sugar-like compounds, namely sugar alcohols, sugar acid, and sugar polymers. And degradation of these molecules lead to the formation of a complex mixture with include hydroxyl acid relevant to the metabolism. So the initial step is slowest of the mechanism is the condensation of two formaldehyde to form glycolaldehyde. This induction period is set up when the trace amount of glycolaldehyde or higher weight sugars are present in solution. And the Formose reaction requires higher temperatures, basic conditions, and catalysis. So the relevance of the Formose reaction for abiogenesis have been questioned, given that if not stopped quickly, it results in the degradation and polymerization of the sugars, forming insoluble tau. And moreover, ribose is generated in a relatively low yield among a large assortment of other side products, including ribose isomers. So notably, the presence of inorganic reactants, namely borate, and to a much lesser extent, silicate, have been shown to stabilize the cis diols, including pentoses like ribose. Alternatively, specific reagents present within the solution could have sequenced ribose. And then the cyanide polymerization, the hydrogen cyanide is a weak acid. So it can utilize the polymerization to generate hydroxyl acid, amino acid, and N-heterocycles. Sufficiently high HCN concentrations would be difficult to achieve on early Earth, especially considering the high reactivity of the cyanide ion with aldehyde. Solution containing lower concentration undergo hydrolysis to form formal amide and formic acid, pendulum by which cyanide ion may have been concentrated, including freezing of dilute cyanide solution or precipitation over time when dissolved metals and subsequent release. So HCN or product of HCN polymerization may have also been delivered by comets and meteorites. And reaction involving free radical typically have low to no activation energy. Consequently, radicals can generate a variety of products, including polymers from otherwise stable, less reactive molecules, for example, carbon dioxide, nitrogen. And then the classic Phaeton reaction describes the generation of hydroxyl radicals during the oxidation of ferrous iron by hydrogen peroxide. One form, the hydroxyl radicals oxidize inorganic ions and organic molecules. Similar process could have occurred on prebiotic Earth, given that its oceans were enriched in ferrous iron. And Stanley Miller and Harold Urey, they utilize this spark discharge experiment. Mr. Gan has introduced this mechanism just now. So discussion of these organics into an aqueous solution can lead to subsequent reactions such as HCN polymerization and the Strecker synthesis. And the polymerization from simple building block of organic monomers to complex biological macromolecules represent a critical step in the emergence of life. These macromolecules, including proteins, nucleic acid, polysaccharide, or analogs of them. The synthesis of protein and peptide polymers is a particularly challenging process under prebiotic conditions. Because the formation of peptide requires undergoing dehydration condensation, which is thermodynamically unfavorable in an aqueous solution. So studies in simulated earlier scenario into intercellular environment demonstrate various pathways for polymerizing amino acids from peptides. Including the hydrothermal wind, the wet-dry cycle, the freeze-melt cycle, mineral surface catalysis. So the formation of prebiotic peptides may provide primitive catalytic activities that will potentially accelerate other prebiotic reactions. Nucleic acid, RNA, or DNA are another kind of core biomolecules in the current life. As for amino acid, the polymerization of nucleotide in aqueous solution is also unfavorable, thermodynamically unfavorable. So the biological synthesis of nucleic acid requires template, enzyme, and a series of cofactors. And the reactant for condensation reaction are always NTP nucleotide triphosphate, such as such synthesis process would be impossible under prebiotic scenarios. So researchers have revealed some plausible routes to template-free polymerization under prebiotic conditions. It is also possible that alternative nucleic acid systems with a simpler structure than RNA or DNA serve as evolutionary precursors, including peptide nucleic acid (PN) and serose nucleic acid (TN), which are simpler synthetic pathways might have reduced the activation energy barrier. It's worth questioning, however, whether these alternative molecules are capable of efficient information transfer and self-replication required for the evolution of life. So modern life uses homocarrier sugars and amino acid. Counterpart remains an active area of the research. So mixed chirality polymers are less functional and in some cases less stable, but they may have been sufficient for early life. Alternatively, the two enantiomers could have been originally served different functions. An organism may have evolved to utilize only one enantiomer or more efficient reactions. However, too much enantiomer diversity could could happen polymerization and enzyme activity. So numerous abiotic mechanisms for promoting hormone chirality have been demonstrated, including the preferential catalysis, crystallization, and adsorption. So the informal polymer used by extant life as a nucleic acid, DNA, and RNA, and classification of how the information in DNA is converted into the function protein came with the discovery of messenger RNA and transfer RNA with the discovering of the genetic code. So codons are the key to the translation of genetic information, directly the synthesis of specific proteins. But the origin of first information regulation is still a mystery. And now I will introduce some significance of nitrogen amino acid AMP. So using a natural amino acid, nucleoside, and triphosphate as a reactant, the synthesis of prebiotic peptide was successfully achieved. The study revealed that the key intermediate N-amino acid NMP were generated during this reaction. So further analysis indicates that the presence of different nucleotides in the reaction system significantly influenced the peptide formation efficiency of the same amino acid. So using this product, using these molecules and amino acid AMP as a primitive tRNA model, we simulated the early Earth aqueous environment by subjecting racemic amino acid and nucleoside to chemical reactions in a triphosphate containing aqueous solution. The result indicates that during NAAMP formation, amino acid and nucleoside exhibit chiral butyl selection. When D conformation nucleobase used by early organisms is present, the L conformation amino acid is selected. So the origin of prebiotic synthesis, hormochirality, and genetic code we discussed before, the three cornerstones problem in the study of life's origin. So I'd like to leave you with the following idea. We hypothesize that this synthesis was not merely a step in the forming a material building block, but also played a critical role in concurrently driving the selection of informational bearing property, named molecular chirality and the foundation rules of codon amino acid pairing. We refer to this integrated framework as a chemical information co-evolution, CICE. So the core principle of CICE is that the synthesis of matter and the regulation of information were not sequential or independent events, but rather two phases in a single intervened process. Under this model, the synthesis of matter is investigated by simulating plausible early Earth conditions, such as fluctuating pH, temperature, mineral catalysis, radiations, and wet-dry cycles to drive the formation of organic compound. So CICE persists that prebiotic chemistry, much like an evolving language, was not simply generating a random vocabulary of molecules, but was simultaneously writing the grammatical rules that govern their meaningful interactions. By adopting this unified perspective, we aim to provide a deeper understanding of the intricate process that govern the chemical evolution of life. And that brings me to the end of this presentation. Thank you for your patience. There's one report left, and now we have a 5 minutes break.
क्वेश्चन है तो अपने घर रख के आना था क्वेश्चन था आपके पास तो अपने घर रख के लेके आना तो लेके लेकिन उनको बच्चों को बताना चाहिए अगर कुछ सही समझ आ रहा तो आपसे पूछ लो वो दोबारा बेसिक से देखिए आपने सवाल भी करना समझना है तो फ्री हुआ नहीं बहुत अच्छा रहता है इसका स्टैमिना अच्छा था आप हमें बताए ना कि लेकिन आप हमें वो कहता मैं उन्हें क्या मैं Thank you so much. Very good.
Yeah, your English is very good. Excellent.
Yeah. इसे पिछू करके आया Take
Oh, yes. 3 years. Everywhere. Yeah, that's okay. Yeah.
मैं भी यही कह रही हूँ इसका कसूर जो कहा गया है उन्होंने वही कहा
That's bigger picture.
If you are doing your master in bio, we have the professor of biomedicine.
You are in
Who's going to take picture? Okay, she's.
I'm not sure to do it. Okay, ma.
You are listen.
Yes.
Maybe we can call our join also join. First take. Okay, fine.
Okay. Mama.
Join us. Yeah.
大家一起来拍一下照好了。
别说啥啥呀。 Ganda.
Ask your friends to come. You can sit in.
Sit, 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 sit.
Okay, come. When do you need? Sit.
बैठ जाते हैं
好的，好的，好的，你的手就好好。一起拍。好，慢一点。好，好。
He's taking picture from here and there can ask them to find picture.
You need to take it, apply, and yes.
像
共享一下。
我先来把它打开，
先打开再共享。
control 一下。
सीक्वेंस अच्छा तो उसकी सच्ची बची लेकिन
你直接打开吧，直接打开，看行不行？
刚才没有复制上，哎，不行啊，
直接打开。对对。
可以共享是吧？
对，通过这个共享，还有点这个我也看懂。
这个是吧？
对，好。可能没有共享。
这个。
可以那个。OK，
这应该共享吗？
对。哎，但是那 OK。那个。
So here are the fluorescent.
这个是哪个？哦，这个就在这里边了是吧？可以了啊。你要说一下吗？
上面不知道共享没有？你看上面怎么没有那种？
这个共享屏幕，
是共享
共享屏幕，对吧？
ये immediate.
OK。
So afternoon, so this part I think will be the last topic of this afternoon. So I will make it I think as quickly. I think most of you may be tired of that. Okay, so my topic is about the fluorescent probes for bioemitting. There's a little difference between just with the two other topics. So let me introduce myself. I'm Jin Hui, one from the IDDT. Yeah, actually, I graduated from in France in 2018, and come here now. It's my 7th year in IDDT, and my major is also about the fluorescent probe. So it's about organic synthesis and the fluorescence imaging. So what's the use of this? So we'll discuss in later. Okay. So at the beginning, what is the luminescence? It was explained that with ESANC originated from the Latin word means to see, all physical, chemical, and biological process described with sense are perceptible by our eyes. So firstly, it's added to see with our eyes. And luminescence refers to a series of light-emitting phenomena, for example, but it's different from the like lamps or sunset because it produces or generates some heat or large amount of heat during the emission process. But luminescence, when we use this word, it means will not produce a lot of energy or heat during this process. Like, photoluminescence and fluorescence luminescence. For your, I will use this. Okay, electroluminescence, chemiluminescence, and bioluminescence. So what are the difference between them? So let's see a picture. Could you please tell me which type of the following emission belongs to? So first, this, maybe we can see it still in somewhere in the forest or some in night, maybe we can still see it somewhere. But here it's not very often in city life. So this, yeah, from the insects, so we call this bioluminescence. How about this? It's about chemical compounds. And the color, we can see the color by irradiation of under the UV light. So we call this is photoluminescence, also including the fluorescence or phosphorescence. And about this, in some in some activity, when a female singer comes to a city, with this, okay, you're right. So what is this emission belongs to? Chemiluminescence, because it has two kinds of chemicals. When it comes together with oxidants and the chemicals, when it oxides to the other state, so it becomes luminescence. It's called chemiluminescence. And this, a jewelry, in old times, a king or kingdom or a princess like to have this kind of what called this, and it's also photoluminescence. It's phosphorescence. Without light, it can still emit for a period of time. So let's continue. How does photoluminescence origin? We'll concentrate on photoluminescence. How does it origin? There's a famous diagram called the Jablonski diagram for fluorescence and phosphorescence. So we only see the right part. When electron was in the ground state, when they receive a light, they will go to the singlet state or the first singlet or the second singlet state. So it means it was irradiated. After the light was removed, usually the energy will lose and goes back. There are two kinds of process. One is it goes back directly to the ground state. This is called fluorescence. But there's another situation. The singlet state has the inter-system crossing to the triplet state. So there is energy loss like non-irradiation energy loss. And the triplet state goes back, when it goes back to the ground state, we call it fluorescence. The difference between the fluorescence and the phosphorescence is their lifetime. The fluorescence has a shorter lifetime than phosphorescence. The phosphorescence usually has like a microsecond or even less, or even longer. So that's why we call it, when we remove the light, we can still can see the still emit in this jewelry or in this part. But the fluorescence, when you remove the light, it will not emit, so we can see nothing. And there are two major concepts in the photoluminescence. One is excitation, one is emission. The difference between the center of these two peaks called Stokes shift. So this is the original idea of this photoluminescence. Now we continue. After photoluminescence, we concentrate. My work is concentrate on the fluorescence. So we designed some fluorescent probes. So what is it for? We use it for imaging in biological process, like disease or some in some environment, for example, as a test paper, blah, blah, okay. So fluorescent probes are molecules that absorb light in a specific wavelengths and emit light of different typical longer wavelengths and process known as fluorescence. It means, according to the lambda equals HV, when it has ground state to the singulated state, when it goes back to the ground state, it's called fluorescence. It will has energy loss. Less energy means longer wavelengths. Okay, so our fluorescent probe, it means has the longer wavelengths than the absorption wavelength. Okay, so that is the principle of our design strategy. We will use a fluorophore. Yeah, usually it's in red color. It is has lower background in biological body. Yeah, for example, in animals, in cells, or in even people's organs. So with a linker, usually this link is kind of microenvironment active. For example, in cancer cells or some inflammation in diseases, there are some enzyme or some ROS reactive oxygen species or some GSH, for example. All these kind of things grew, increased or overexpressed in this in body or in cells. So how to imaging, how to detect? There are many methods. For example, usually we go to the hospital, we check by, for example, fluorescence or the immune system. And if we use the fluorescent probe, it means we can use this technology there without invasion. For example, when you have some blood cancer, usually they go to the hospital with like kind of mirror, but it's a very tiny mirror, goes back to your body and checked on the machine to see what's wrong with places and what's the injury part. But if we use the fluorescent probe, it gives us a kind of non-invasion technology. So this is the receptor to target the place, for example, in cells or in specific organs. Okay, this picture shows us the normally developed fluorophores, starting from, you can see the wavelengths in blue to near infrared. For example, here, for example, body B or rhodamine fluorophore, it has the kind of wavelengths at around 600. This part, for example, it shows orange or yellow. And if we want to get a better fluorophore, it means we should use this fluorophore in a body without or with less background or noises from the body or microenvironment. So the scientists prefer to choose in red emission part. It means more than 700 emission at this region. These are some kinds of mechanism during design for the fluorescent probe. It included photo-induced electron transfer like PET or ESI, PET, FIT, and ICT. Okay, this is the strategy to design the fluorescent probe. And what kind of fluorophore we use, it depends on our reality. For example, it depends on the wavelength to increase the conjugation. For example, one benzyl group, you add another benzyl group or alkyne or this kind of conjugate, pi conjugate group. So we could increase the emission wavelength to near infrared or near infrared 2 regions even. Okay, these are some application of the fluorescent in diagnosis and therapy. For example, in the detection of biomarker for disease, just as we discussed for some biomarker of this kind of disease, cancer, inflammation, or any kind of disease. Usually they take some with blood or use of the urine. Okay. And to kind of also we can use it for the drug delivery. Some fluorescent probe we can to see where is the drug exactly is. For example, how did the drug like metabolized in the body. Where it is, it's in the liver, in the heart, or it goes to the brain. So that we can use the fluorophore to like detect where it is. Also, it can be used for the early diagnosis and therapy of cancer. Here are some examples of using the fluorescent probes. For example, the first are in cells. Use the fluorescent probes to imaging in cells. For example, here there are different colors, for example, from blue, green to red even. And this is these two pictures are used, are fluorophore used in real hospital on real patient. For example, here is the yellow 560. This is it emit orange color or yellow color. It calls yellow color. They use this to imaging when they do the surgery. We inject this kind of fluorescent probes. So it in the cancer part or cancer tissues, it emits red. So the doctor could like to remove this cancer part by this fluorescent probe. This is also called ICG. ICG is the first FDA approved fluorescent imaging agent. It's already like more than 1 million surgery in the world every year. Use this kind of ICG. Yeah. Even in Chinese hospital in Ningbo hospital, they use it more than around 100 surgery per year. Yes. Okay. And here is the zebrafish. We use the fluorescent probes to imaging the zebrafish. You see where it located, like in which part. Here is the control group. This is a diseased model, zebrafish model. And how to develop this kind of fluorescent probe? We just talked about we use a fluorophore and we use the linker. And if we want to use it for therapy, we can link with a drug. For example, use a cancer treatment drug and with a kind of ligand as the target and the fluorophore as the imaging or sensitive group of fluorophore. And this linker is the active part. So many kinds of papers have been published. Even in our group, we have developed some of this kind of fluorescent probes based on this principle. So these are typical three developing strategy. With the first is for diagnosis. With the target ligand, as we said in the beginning, with the target ligand and linked to the fluorophore. When they target to a specific cells or tissues, we remove the target ligand and release the fluorophore so we can see where it is. And the second is for therapy. When we link it to a drug, also here we have the fluorophore here, with a drug in a specific cell microenvironment, the target ligand will release by the linker reactive. So this drug is for treatment. This third one is the theranostic fluorescent probe. This fluorophore could be near infrared or green or yellow even, whatever. We can use it for like a combine of these two, diagnosis and treatment. In this case, we can use it for target disease region or in a cells or in a specific microenvironment active ones. And for fluorescent imaging as well as the drug release. So this is a theranostic fluorescent probe design. And our group has worked on this in this like 7 years. And our group have developed some of these three kinds of fluorescent probes. One is the organelle target fluorescent probes. The second is about some small molecular like GSH or ROS reactive fluorescent probe. And the third is about enzyme, like amino peptide donases and neutral reductases and beta GL responsive fluorescent probes. So first we will start from the organelle target fluorescent probes. So we begin from the lysosome. Lysosome is one kind of called the digestive organelles in the cell. It is organelle in eukaryotic cells. It contains like various kinds of hydrolytic enzyme. It also called the digest factory of the cell. It can digest many kinds of the small molecular or even large proteins inside these organelles. And it can break down, vagrant endogenous or exogenous micromolecules. When it has dysfunction, some disease comes to us. For example, the lung diseases, the lithium storage disease, like also called LSDs. This kind of type 2 chloride storage, for example, and also about tumors and various other diseases like inflammation. The lithium has a very typical environment. It has acid, less or lower pH. For example, normally for our body or cells, the pH is about 6.8 to 7.2. Yes, but in lithium, it's in an acidic environment. The pH can lower to be 4.8, around 5. So it's like the digest in our stomach. Yeah. So first we designed this kind of lithium targeted photo synthesizer. This is a structure. So we use this part as it's also called a kind of MI. It's a kind of lithium targeting group and combine. Yeah, I will increase this at the next slides. So we can use this for lithium imaging as well as the photodynamic therapy. This is a molecular structure. This part is for the lithium targeting. And this part also called TBEDH is a fluorophore, but also a photosynthesizer. What is a photosynthesizer? It is kind of molecular that with irradiation by the light or a laser, for example, because with a specific wavelength, it can produce some reactive oxygen. Yeah. So also called ROS. This ROS is kind of like it's very important for the body like equilibrium, but when it increased, it will damage the cells. So we use this. We use this to increase the ROS in cancer cells. When it increases, the cancer will broken. So this is kind of a kind of treatment. It's very often, especially for the, this kind of photosynthesis is already in medical use, especially for kind of skin cancer. Yeah, we inject with kind of photosynthesis and under the irradiation of a specific laser, and this cancer cells will be broken and be cured by this kind of photosynthesizer. And also we use the DFT calculation to have a better understanding of its conformation with the normal hemo. This is the normal properties, normal like characterization. And to see well about these properties, this probe has very well AIE properties. This is called aggregation-induced emission. Normally for the fluorophore, when it's in the aggregate solution or aggregate in solid or in high concentration, usually the light was the photobleaching. But some molecules has a very special properties. When it in aggregates, it has high emission. So what's called aggregate-induced emission. So this compound has a very typical AI properties that also give it special properties of the photosynthesizer to increase or induce the ROS in the body. And also we checked in different pH and other interference. It has very good anti-interference ability. Also in the photostability as well as the biosafety. So before applied into the animals or even real body, we should first investigate their biosafety. For example, we use kind of in cells or in tissues, for example, or in the organs to see how to check with our design, our original design is to target the lysosome. So we have to compare with other organelles. For example, we use this probe to compare with the lysosome and mitochondrion and lysosome and mito and the lysosome. So it has been found that it has a very good Pearson coefficient around 1.97. So 1 is totally co-localized. So the close to 1, the best of the co-localization. It means a very good co-localization compared to the other 2 organs. One is 0.6 and the other is 0.41. Also, we checked. It has the more red color of this part, this part especially here, the more red color, it means it produces more of the ROS, so that it can kill or produce or broke the cancer cells in this part. And this is about the ROS production in the tumor parts of the tissues. Again, we just talk about the lysosome. How about the other organelles? It's called mitochondria. This is the structure of this kind of organelles. These are very common organelles in eukaryotic organelles that make chemical energy. It's called the energy of the cell. It produces ATP. ATP in these organelles. And the dysfunction of this also involves a lot of diseases, the kind of tumor or related, mito-related diseases. So we use a similar strategy. We use this kind of probe to add a kind of mito target group. That's called the PPS, the phosphorus group, this part, here part. PPH3 as this kind of target unit. We also have involved the photodynamic therapy in this part. It produces ROS. It can convert the oxygen to the high active oxygen species. It's kind of a radio oxygen. And continue with that, we also developed some enzyme reactive or responsive probes. So what is the nitroreductase? What is the function of that? It can produce or reduce the nitro group into 1, 2, 3 steps to the amide. This is a very important property in the cells. It can reduce the nitro aromatics to the corresponding amine aromatics. And it's also a biomarker of the hypoxia. So what is hypoxia? It means less oxygen. Usually in tumors, it grows very, very fast. Its proliferation in very short time, it can proliferate in many times than the normal cells. So in this case, the oxygen is not enough. It's not enough. So we call this is hypoxia. It with less oxygen. For example, in normal tissues, we have 5% or 20% oxygen. When we breathe with 20% or 21% of oxygen. But in these cancer cells, it can survive in less than 5% of the oxygen. It grows very fast, so it needs less oxygen. It can survive in the hypoxic condition. And this NTR, also called, it means nitroreductase, is a biomarker of the hypoxia. So especially it's a reductase, so it can survive in the less oxygen. So this is a biomarker of hypoxia. So if we can detect the NTR, it means that we can detect the tumor in the early stage. in hypoxia situation. And it's overexpressed in primary tumors. Now here is the developing fluorescent probes of our NTR reactive fluorophore. So at the beginning, it's fluoro turn off with the mechanism we called ICT, intercharge transfer. With this moiety, it's like a non-fluorescent. But in presence of the NTR or nitro reductase, this part was reducted. Nitro part, NO2, was reduced to NH2. So in this case, NO2 is a very strong accepting electrons, but it goes to NH2, it changes to donating part. Okay, so then this is in the electron was changed. So the fluorophore changed either, also changed too. And it shows near inflorescence. From non-inflorescence to inflorescence, it gives us a kind of opinion to imaging the NTR in the tumor hypoxic cells. And we also, this is the NTR probes. We use this fluorophore with NPH3 part, and with the 2CN as the accepting groups. And this is for lithium targeting. And here is the nitro reductase active part. So in this case, we can use to like this probe can be used for ratio matching from the red fluorescence to green fluorescence with two color different change. If the color changed from red to green, it means it has overexpressed of NTR in these cells. Continue. We use not only for imaging, but we also want to develop some fluorescent probe for therapy. And this is the fluorescent probes we design for therapy. This is OS reactive with in this linker. It's called the OS linker. In presence of larger amount of the OS, this part will be broken and release the drug. This is called CPT. This is a normal cancer drugs. It's like it can cure a lot of cancers. The mechanism of it is to conjugate with the DNA and breaks this part and go through this. This is for chemotherapy, as well as this fluorophore can also be used for the photodynamic therapy. It means it can produce larger amount of excess of oxygen or high reactive oxygen species. So this we can combine the diagnosis imaging as well as for the therapy, for cancer therapy, chemotherapy, and PDD or photodynamic therapy, especially for these lymphoma cancers. Also, we developed what is reaction oxygen species we have just talked about. It's a kind of high reactive oxygen like HL2O, OH, like epoxidine, nitrogen oxide, and this species. When it's overexpressed in the cells, it can destroy the cell protein, or lipid or DNA even, DNA, and DNA damage or lipid oxidation, as well as induce the cell apoptosis. And it's also associated with various diseases like aging, cancer, and neurodegenerative diseases, or cardiovascular diseases, and diabetes. So it's oxygen stress related to many kinds of diseases, like from the cancer to the brain related. Okay. And this is our recent work. It's the for oxygen ONO, concentrated development fluorescent probes. This fluoride very interesting is that this fluoride is kind of locked in the normal state or in the without the instimulation of the oxy-ROS. So it's like fluorescence off. But in low ROS with condition, it releases this part of broad part. So to release this kind of near infrared fluorescence. So at this here, it's like no fluorescence, and in low concentration of RS, it releases this part to emit red fluorescence. And continue if the ROS increased, like in high concentration, this part, this carbon bond will be also continue to be broken. In called this part is the release of this part, or we call our XOH part, and this emit yellow fluorescence, a very strong yellow fluorescence. So the in vivo experiment also prove this this phenomenon. It's called a kind of cascade reaction with different concentration of ROS. So this is a dual channel technique, and this is already submitted, and we will get the feedback soon, I think. So as a summary, what will be the next? We want to develop the near infrared fluorescence, and it can increase the penetration depth. So the longer wavelengths, it can penetrate deeper. Yes, but without the background noises. And we also want to have the inner and outer modification of ferritin, for example, to transfer or deliver the fluorophore of the probes to the targeted cell or cancers or inflammation part or tissues. And also we will use it for in vitro and in vivo, treating the tumor markers. And what we are doing now is we are using it to detect the urine of humans, outside the body, to see what's the biomarker of the patient or compared to the normal ones. And we develop kind of APP, yes. We will wanted to use it in the hospital and to make it true in the reality. And for the normal people, like at home, you can check with what kind of cancer or a specific cancer, especially for some kind of people has a kind of chronic diseases. We want to check what's the level of this kind of biomarker. So that is our future work. And this is to use the AI detection with the body fluid, not only for the urine, also rather like sweat, for example. And your tears can also be a detect our tool for the detection part. Yeah, okay. So it has increased, for example, the enzyme or the ROS, for example, any biomarker, it could be detected by the fluorophore. Just use the color, compare the color change. Okay. Now at the end, thank you. And I'm looking forward for the cooperation of our colleagues. No, you are colleagues now. Yeah, we are we are colleagues now. Okay. So if you want to have some questions, please. Okay.
So you are using folic acid receptor only to target the cancer cells?
For example, sorry, yeah.
You are using only folic acid receptors to target the cell? You are using ferritin for target?
Yes, yes.
So they will target the folic acid receptor on the cancer cells?
Yes, yes.
So why you are not using the transferrin receptors?
Actually, you mean the ligand, yes.
As a ligand transferrin, and they will target the transferrin receptors. Again, they are doing the same function. They are delivering iron to the growing cells.
It's also a good idea, yes. The how to say that, the ligand or the target, yes, receptors. So that's what I wanted to develop to our delivery. We use the ferritin for just as an example, because we have a collaboration with the ferritin produce group. We can also use what you have said.
Basically, I am also working on the theranostic nano.
Yeah, that will.
And I have already targeted folic acid receptor and transferrin. These are my big patents of to theranostic nanomedicine.
Yeah,
that's as a fluorescent. I am using the nano vehicles, quantum dots.
Quantum dots.
Quantum dots. I think you are using the dyes.
Yes.
Like FITC and methylene, danger, Texas red. These are the dyes that you are using as a fluorophore.
Yes.
But I am using the quantum dots.
Quantum dots.
And the quantum dot as a vehicle for targeted delivery.
how was the quantum dots? It has like a reactive, responsive, or change light? No.
Basically, quantum dot just for as a delivery vehicle.
Yes.
We attach the linkers on the quantum dot,
Yes.
and then on the arms of the ligand, we attach our drug, our
Yeah.
ligands, and engineered those nanoparticles. So the size of those nanoparticles are below 10 nanometers. That's why they are known as quantum dots.
Yes. Yeah.
And when we completed our formulations, the
Mhm.
size is maximum, I think, below 100 nanometers, the final size of the formulation. So
Mm.
the delivery, their clearance is very good as compared to fluorophore.
Okay, that's good. In this case, the main drawbacks of the fluorophore is that it has no target.
Yes.
Yeah, so we should incorporate a kind of delivery system, as you said, condoms or other delivery system.
Even
Yeah.
in that case, you can
Yeah.
design the bond property,
Yeah.
the bond that can break in the hypoxia condition at lower pH and
Yes.
deliver the drug more efficiently. Even you can do the passive targeting by
Yes.
using that.
Exactly. That's what we wanted to develop next for use a kind of like a delivery system for a shell, make a shell or more for nanoparts or other kind of nanomaterials to develop or combine or like make a shell of our fluorescent for inside, for example, like goes directly to the disease region or tissue or cells even for anywhere.
But
So
we are specifically
yeah.
working on the specific targets. Every
Mhm.
cancer has its
What kind of
specific
targets do
target.
you use? Receptors,
Sometimes CD regions.
CD
There are
region?
specific CDs,
CDs?
specific type of cancer,
Yes.
like CD122 for liver cancer,
CDU and okay.
for renal cancer. So we are designing different type of theranostic
Mhm.
medicines against different cancers. And mostly our linkers are biological linkers. So
Okay.
our body have a system to metabolize those
Yes.
linkers too.
What do
So
we
as a
want?
biochemist,
Yeah,
basically I am also a biochemist.
that's good. It's a good collaboration
certain
part
reasons.
between the chemical chemist and the biologist.
Yeah.
Yes. And what I'm wanted to use the ferritin. Ferritin is a kind of target the TFR1 receptors. Yeah, it's also kind of and this because we only have this. Okay, and it will be a good collaboration with the different kinds of nano materials and with different targeting receptors on the cell surface of the tissue part. Yeah, we can talk more with this. Okay, good. Okay, okay. Yeah, you can come. We can discuss. Yeah.
Can
Oh,
we use it for
Yeah.
DNA content?
No, it's okay, no problem. Can we use a DNA, small DNA,
Yeah, no problem. They're kind of
500 or 700 BP? Can we
Yeah, no.
combine it together and we can use it checking in the chromosomes? Is okay?
checking use DNA as
Yeah,
the
use a DNA,
yeah.
but checking in a chromosome fluorescence. After that, checking this is contained from the father or mother. How
I
the
think it could. I
hybridization?
think someone has already done this maybe.
But we have a special DNA probe.
Oh, you different special.
So we should make a probe with the fluorescence. How we can do it?
Do you want to make a okay? Do you want to make it like a very selective or you can what kind of fluorophore do you want
For the fish?
for the fish? For
Yeah,
the fish.
for fish.
I think we have already, as you have said, many FRTC, ICG, for example, this kind of fluorophore has already been used.
But this is a ribosomal genes.
Ribosomal,
Yeah.
okay.
This is a difference.
Different. I
What
think
is liposomal?
ITS gene names, you know?
ribosomal. Okay.
It's different taxonomic genes.
Yeah.
But
Yeah.
you just have to conjugate that probe with the fluorophore.
Yeah.
I think target probe.
This is a target is for understanding which parents can be.
We have a probe that have a complementary sequence against
Yeah.
a specific region. You already have a probe.
Yeah, hybrid system.
That is always a probe is complementary to certain region of a DNA.
Not sorry, what again?
Just like when we are the real-time PCR. In real-time PCR, we have a Tachman probe.
Yeah,
In southern blotting.
yeah.
In southern blotting, you also have a probe. The
Uh-huh.
probe is a specific complementary region against a specific region of a DNA.
Yeah.
So if you have a probe, that means probe is going to target that specific region. So it's already targeted. And if you want to trace that probe
Yeah,
So in
we
that-
want to combine this as a fluorescence.
In that
so
case, yani can
No
be
probe. I
conjugated.
mean, this is no probe. Only we have a sequence and marker primers.
Okay.
But how we can combine it, that sequence and marker for that one? After that, we want to check inside of the chromosome. They go the father, how to say, the child or not?
I think for the you already have a sequence. What kind of
Yeah,
sequence? It's a kind of amino
let's
acid or
I try my idea about. We have 3 species. We have that 2 species is maybe is one of the parents of that species, and we don't know how it's a hybridization going, and so we make the probe, DNA probe, not probe. This is only DNA, which is we got 3 species little different, but is primer they can read.
A specific region,
Yeah, I have
polymorphism, that there is a polymorphism in that specific region.
Yeah.
So you can design a primer or smaller nucleotide probe of 10 to 20 nucleotides.
Yeah, we already designed it, but
And
the
if you
fluorescent.
conjugate that
Uh-huh.
with a fluorophore or any quantum dot, and then you deliver it to the fish. If it conjugate, then you get the in vivo imaging via fluorescent bioimager. We have a bioimager. If you put the fish or any animals inside that imager, if the target bind to the target site, it shows there is no polymorphism. The sequence is same as the parents have. And if there is no fluorescence, it shows there is no binding between the
No. We need to make a probe, DNA probe. Just give we should give the primer and how you can contain the fluorescence. This is easy. Not for me, not easy because I'm not working with the fluorescence. But how we can make the probe very easy.
To make it prudent, I think the
probe is
to only
banana.
target this DNA, or it can
Only target this
only
DNA.
target
Yes.
this DNA. That should be only target this DNA. That should be very important issue. I think it's very difficult to only target. What is the difference between this DNA and the others? Yeah,
Okay.
that is the difference with
You,
this.
there is a simple principle.
Yeah.
Yeah.
Just like as we check the polymorphism,
Yeah.
any specific mutation at any specific place of
Yeah.
gene.
Yeah.
So you
You need to use the process.
probe for
Yeah.
that.
Yeah.
You can conjugate that probe with the
Yeah.
Yeah.
fluorophore
Yeah.
and then deliver it to the.
Yeah. So
I have some question.
yeah, please.
So I separate arrows in your cells. You break them.
Yeah.
What effect those high arrows have on the healthy cells? How?
Dose. You mean the dose of
Healthy cells don't have high RS.
They do, they are
It's
they
only in the lysosomal part.
are to break the arrow.
yes.
Okay, they are increasing the concentration of our ROS.
Yes.
How will you tackle that the cells which are around those disease cells, they will remain unaffected?
When the RS was generated because of the laser comes in the part.
Okay,
Specific laser.
we will make sure that it is not going to have effect on.
Yeah,
the
so
nearby cells.
where you like irradiate part is the laser, you irradiate it increase the RS. The other part will not increase because it can only produce this.
How will laser know that
Yeah.
I have to target these cells? There
So
should
the fluorophore.
fluorophore. Just like the gold therapy.
Fluorophore,
case
which
of
means we
magnetic-
have the part.
In case of magnetic nanoparticles and gold, they can trace them.
Yes.
But
Where the-
instead of that, we are the problem that selectivity is still the main issue. Another thing is
Yeah.
where that we are also working on the nitroreductase probe, nitroreductase targeting probe.
Nitroreductase flow. Yeah.
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 So nitroreductase is also present in your healthy cells. And again, you will use the same laser approach to target that
That's the
specific.
difference. So it depends on the fluorescent probes, the detection limit of that one. And the difference, in this case, these are only cells experiment, but in real like cancer or different, there's some difference between the NTR levels. For example, if we can use the fluorescence to quantum or semi-quantum analysis, we'll know what is the level of the NTR in the real
Yeah,
disease part. Yeah.
in the cancer,
Yes.
it will be higher.
Yes.
Yeah. But what about the healthy cells? They will also have,
Yeah.
although they will, the level will be lower.
Yes.
And how will you ensure that only the higher
So
level?
that is, we should use the semi-quantum analysis.
Okay.
Yeah, to see what's the strength or intensity of this emission.
Because
Other than that,
When
you are
the
going with
control
the
group,
glutathione reductase,
Yes.
then that's the main issue because glutathione reductase is in higher concentration in your red blood cells. Because it is the defensive mechanism of the body against reactive oxygen species. Red blood cells are exposed to the oxygen.
Mhm.
That's why the concentration of these free radicals are higher in our red blood cells.
Yes.
Again,
Just
you have some personalization, you have some company.
No.
Tada.
You don't use it for the any minute or something
Yeah.
for the.
These are-
Only you can make by yourself.
Yeah, we didn't take.
Actually, I know some companies.
बस मैं ना अब हमने कहा फिर अपने पास ग्रुप में जाना अपने पास अभी आपके 4 दिन इसी सब्जेक्ट so you have
Because we
biochemistry के हैं so you get the many opportunities to ask that question.
I can check with you.
Yes,
please.
for presentation.
I can check. I can check. Yes.
So
Then have your email. I will check. Okay, you have with that.
then
Okay.
Thank you very much.
Very well.
Thank you. Thank you. Okay.
For now, are there high detection?
Okay, sorry, I said sick. Okay, please, we are finished now. Thank you, everyone, and see you tomorrow. Okay.